Okay, good morning and thank you for joining us. Please be advised that this seminar is being recorded and will be available at a future date on our CASIS website. The Center for the Advancement of Science and Space, CASIS, is manager of the International Space Station National Lab, and we are hosting a 3-day online seminar series focused on tissue engineering and regenerative medicine in space. The seminar series will review key tissue engineering and regenerative medicine research conducted on the ISS National Lab over the past couple of years and examine how the future of this important research could advance biomedical discovery. The series will also discuss specific areas of research and new capabilities that enable life sciences research and in-space production on the ISS. We welcome you to the second of the CASIS seminar series on tissue engineering and regenerative medicine on board the ISS. Today's topic is organoids and microphysiological systems. Normally around this time, we would be gathered in person at the ISS R&D conference, but due to the COVID-19 pandemic, we have moved to a virtual environment. I encourage you to visit our website to learn more about virtual ISS RDC opportunities and to engage with the community. My name is Mark Giulianotti, and I'm a Senior Program Director for CASIS. But as you know, it takes a team of individuals to bring together a seminar series like this, and at this time, I also want to acknowledge the rest of the CASIS team that has worked to bring this series to you. As you will hear in the following presentations, a number of studies on ground and in space have indicated that organoids and microphysiological systems, some of which contain organoids, can be utilized to study human health and disease progression and to improve the attrition rate of therapeutic development. In space flight, humans experience symptoms that appear to be similar to accelerating aging effects. For this reason, organoids and other microphysiological systems have been sent to space to see if they can serve as accelerated disease models for the development of therapeutics to treat patients here on Earth. And today, I have the privilege of briefly introducing you to our speakers, who will each spend 20 minutes presenting their topics. We will then host a panel session that is open for your questions. We look forward to your participation. Please feel free to enter any of your questions in the chat window. Our moderators will collect all the questions and pose them to the speakers during the panel session after the presentations. I also encourage you to visit the website to read more complete biographies of our esteemed speakers, as I only have a few minutes. So we'll start off with the first speaker, Dr. Jean Loring. Jean is a world-renowned stem cell scientist and co-founder of Aspen Neuroscience. She was founding director of the Stem Cell Center in the Center for Regenerative Medicine at the Scripps Research Institute and co-founder of the Stem Cell Center at Sanford Burnham Preb's Discovery Research Institute. She is author of the Human Stem Cell Manual and has an issued patent for PluriTest, a novel bioinformatics test for pluripotency that is publicly available and has been used over 30,000 times globally. And of course, she was part of a team that sent organoids to the ISS. And with that, Jean, the audience is all yours.
Well, thank you, Mark. What I want to talk to you about today is the really exciting work that we've initiated on the ISS. And I'm sure, as all of you understand, we really need to design a different approach in order to ask the questions that we wanted to ask, because putting cells or putting cultures onto the International Space Station requires a lot of thought ahead of time, and interestingly, a lot of trial and error. What we're studying is a neurodegenerative disease, and we were interested in finding out how neurodegenerative disease may progress differently in microgravity than it does on the Earth. The sponsor of this program was the National Stem Cell Foundation. Our collaborators are at New York Stem Cell Foundation and at Space Tango. I'm at Scripps Research and at Aspen Neuroscience. So this is the idea. Both Parkinson's disease and multiple sclerosis are diseases that cause neurodegeneration, and they're characterized by inflammation in the brain. Inflammation in the brain is mediated by microglia, which are the brain's immune cells. So we wanted to create a model of neuroinflammations that we could send up to the space station. We started with induced pluripotent stem cells that we derived from donors who had Parkinson's disease or multiple sclerosis. We differentiated those cells into precursors of dopamine neurons for the Parkinson's model, or for cortical neurons for the multiple sclerosis model. The neuronal precursors were aggregated to form spheroids, which we will now call neural organoids. Microglia were derived separately from the same donors, and then we added those to the matching organoids to create a neural microglial neuroinflammation organoid. We had duplicate cultures of the organoids for about 30 days on the ground and also on the International Space Station. So why were we using neural organoids? We had been studying them for a while in the lab, and one of the really striking effects of growing cells in these three-dimensional structures is that the neurons mature much better in this environment than they do when you culture them in culture dishes. And this is illustrated, at least by one function, by the electrophysiology that was done by Pietro Sana and Irina Sokolova at the Scripps Research Institute. And here you can see in panel C, that they've captured an organoid in a little mesh so that they can attach patch clamp to individual neurons in the organoid. And you can see the outcome is that the neurons are spontaneously bursting, which is a typical characteristic of these cells when they're in their mature state and not when they're immature. This is how we prepared the neural organoids. We used organoids with and without microglia. We started with aggregating the neural precursors, and we used a dish called an agar well, or an aggregation dish. You can see in that diagram, or in that illustration, that the little individual organoids form in each one of the wells. We then added microglia to some of those organoids, and then transferred them individually to cryovials. So each cryovial will hold a single organoid, either with or without microglia. And then they're sealed up, and that is exactly what they'll be in that environment for 30 days, a little bit longer because they have to get to the East Coast from the West Coast, and they have to stay at 4 degrees until they're launched, and then they're turned on 37 degrees to continue to develop. The launch was on December 5th, 2019. This will show you how the aggregation of the incorporation of the microglia and with the neurons takes place. As you can see, there's the organoid. On the bottom are microglia cultured in a culture dish separate, which are very migratory, as you can see. And then on the right, you can see that when they're added to the spheroids, they will start moving into them. So they will create a neural organoid hybrid organoid. This is the preparation of the experiment for launch at the Kennedy Space Station. On the left are Scott Nagel and Jason Stein. Scott is from the New York Stem Cell Foundation, and Jason is from my lab in California. They signed all our names, all the major people involved in it, on this box, which is the powered locker that will keep the cells at 37 degrees over the time of the experiment. And here the locker is on the space station. And you can see that it was not left quite like this, but it's kind of cool to watch it float in space. And cells are inside there in the cryotubes, like that for about a month. we were able to monitor the organoids while they were on this space station. And this is a picture of the tubes live in after 3 weeks, three weeks of the roughly a month. And this may not seem all that impressive to you, but this is one of the most exciting things that I had done in a long time. If you see where the arrow is, you can see that there's a single spheroid, or actually sort of become a little bit flat in microgravity. That this organoid had been in this tube for 3 weeks, and you can see blown up. And it's not great, but it's not great optics, but you can definitely see that that single organoid in that tube is doing just fine. The experiment came back to Earth, and it showed up in my lab on January 8th, 2020. On the left, you can see Jana and Twyman taking pictures of it in my lab, the locker. Twyman is opening it up, and then the rest of us are sort of smiling, and Jason and Twyman and Jana are smiling, and it's in the evening, as you can tell. So we stayed up kind of late that night to be able to see how well the cells did. Here are the organoids taken out of the locker. You can see that all the cryotubes all stuck together. We took some of those organoids, a sample of those organoids out of their tubes and placed them into culture dishes, so we could find out right away within a few days whether they were living and healthy or not. And you can see on the right-hand side, there's the edge of the organoid on the right, and there's a prolific growth of axons out of the organoid. These cells, after they were there in that little tube for 30 days. And then in the lower right, you can see the edge of a culture like that, and you can see the density of the outgrowth. These cells were ready to grow axons. They'd already grown axons within the organoids, and they spread them out when they had an attached substrate. So I'm going to sort of wrap it up there, because as I think all of you are experiencing, we're sort of in suspended animation right now. The organoids are being analyzed by both morphological methods and by gene expression profiling. We saw that both the ground control and the spaceflight organoids survived and they remained intact, and we also saw that they matured. Morphological analysis was delayed by the shutdown, and it's in progress at the New York Stem Cell Foundation. And the gene expression analysis, which was also delayed, is complete, but the bioinformatic analysis was delayed, and we're doing that now in my lab. You can see on the right immunocytochemistry for nestin and N-cadherin, which are markers of neurons, of early stage neurons. You can see that the ground control and the space flight look very similar. The neurons have formed rosettes around a core, and this is reminiscent of the early stages of neural development in the embryo. And then the neurons mature from that structure and move out into the periphery. Still to come, we plan to repeat this experiment because we've learned a lot from what we've done so far, but not quite enough. We're going to be asking whether microgravity affects neuroinflammation, which is characteristic of neurodegenerative disease in both Parkinson's disease and multiple sclerosis. I want to thank everybody, especially Paula Grisanti and Jana Studemeyer and Valentina Fossati, who were my partners in this enterprise, along with Andres Bratlial in my lab and all the rest. As you can see, one of the most exciting things about putting an experiment on the space station is getting to watch the Dragon rocket take off. Thank you.
All right, thank you very much, Jean. Again, we'll hold all the questions till the panel session at the end. So I see that some people are using the questions feature as well as the chat feature, and we'll collect questions from both. So Whichever form you'd like to submit your questions in, we'll maintain that and ask those to the panelists at the end. So next we have Dr. Dioko Kim. Dr. Kim is an associate professor at Johns Hopkins. His group's research spans the disciplinary boundaries between nanotechnology, biomaterials, and mechanobiology, with an emphasis on their applications to tissue engineering and regenerative medicine. Like Dr. Loring, Dr. Kim is also an entrepreneur, having founded Nanosurface Biomedical, which is now Curie Bio. They're focused on integrating human iPSC-derived cells, tissue-specific biosystems, AI-enabled data analytics to accelerate the discovery of new therapeutics. Recently, though, Dr. Kim's team sent their engineered heart tissue platform to the ISS. Dioko, please take it away.
Okay. It is my great pleasure to introduce our tissue chip in space project. My name is Dr. Kim. I am currently associate professor in the Department of Biomedical Engineering here at Johns Hopkins University. So today I will be giving an update on our heart tissue chip in space project and our recent SpaceX 20 launch experiment. Next. As you know, space flight affects almost every physiological system in our human body, including the cardiovascular system. NASA has identified several cardiovascular risks as the area of the high priority for research, including diminished function, occurrence of the serious cardiac arrhythmia, and impaired response to stress and exercise. While we have gained the important insight from the previous study, a shift to using the 3D human in vitro model of the cardiac tissue would significantly help to better understand microgravity effect on the human heart. Further, the findings from this study on microgravity might help inform research on effect of aging here on the Earth. Next. Main goal of our project is to develop the high-throughput microphysiological model of the human myocardium, which is derived from the human-induced proliferative stem cell here in this schematic illustration, so that we can study the effect of the microgravity on the human cardiac tissue structure and function. We have been working on this project over the past 2 years, sponsored by NIH and CASES. And I will be primarily talking about the major progress made during the first phase of our project over the past 2 years, which is to design and validate our micro cardiac tissue model on the Earth, and then send it into the space for 1 month stay on the International Space Station, where their contractile function will be assessed in real time before they retrieved for structural, biochemical, and genomic analysis. Next. So here is our engineered human heart tissue, which is also called as EHT. So we use the EHT platform, which is composed of the iPS-derived cardiomyocyte and the stromal cells that are seated within the hydrogel scaffold, and then cast around the 2 silicon posts, in which deflection of the flexible post here allowed us to measure the contractile force generated by this tissue. You can see the movie of the beating microcardiac tissue within our 24 EHT platform here. Next. Since one of the major roadblocks to using the human iPS-derived cardiomyocyte and tissue for in vitro study is phenotypic immaturization, post-differentiation. We have utilized the novel electroconductive hybrid hydrogel to serve as a scaffold to improve the maturity of the engineered heart tissue. This hydrogel is composed of the decellularized extracellular matrices shown here upper left panel, which is derived from the porcine left ventricular myocardium, and then reduced graphene oxide from the heart tissue with the electroconductivity and stiffness of the RGO. We were able to generate the tissue with enhanced functional maturization. Next. As indicated by the increased twitching force and upregulation of the key cardiac gene and improved the electrophysiological function. Next. We further modified our engineered hard tissue platform by incorporating the magnet into the flexible force. This magnet, when coupled with the array of the giant magnetoresistive or GMR sensor, allow for the measurement of the post deflection in automated and real-time manner. Next, as posts are deflected, the resulting change in the magnetic field is also detected by the sensor, and this can be converted into the voltage signal, then force readout without using the relatively inefficient method of the live tissue imaging by combining the direct sensing here and reference sensing circuit on the board. We are able to subtract out the noise signals to give the clean readout. Next, EHT array and next incorporate into the space flight chamber. They are designed by the BioServe Space Technology, our instrumentation partner. This chamber consists of the hard polymer body, gas permeable PDMS membrane, and self-sealing the pulse for the fluidic exchange. Array of these 6 tissue each can be inserted and the whole chamber sealed to prevent leakage with the end result looking something like this in this slide. Next, and this chamber are next integrated into the BioServe plate habitat or PHAB. For this experiment, each PHAB holds 4 chambers, with each chamber pair residing over the printed circuit board containing the magnetic sensor, seen here in this slide. Once chambers are inserted and secured, the entire assembly is sealed in the anodized aluminum case, then contain the band that allow for the gas exchange when the PHAB is situated in the civil incubator that is already in the International Space Station. Next. So this is what integrated PHAB looks like. Next. And here is the Jonathan Sui, my postdoctoral scientist, who is holding the one up for the inspection while at the Kennedy Space Center. And finally, this picture on the right partially demonstrates how PHAB is loaded into the aluminum casing. For this first flight, we used 2 PHAB and 8 chamber in total. Next. So here is our experimental design. Upon arrival at the International Space Station, all tissue are fed, and then subset of the sample are terminated at the day 7, 14, and 28. Termination consists of the fixing in PFA for immunostaining or preservation for RNA later or RNA sequencing analysis. And subset of tissue will be also returned viable and cultured for additional week on the Earth to see if there's any recovery effect. Next. With the engineered hard tissue platform and the space flight hardware developed, we also conducted the experiment validating testing or EVT. So this slide shows our EVT assessment result. So we assessed the entire system and procedure, and this assessment included the long-term tissue viability and validation of the electronics and our software, and chamber integrity during the freezing with the RNA later, and the quality of the resulting immunostaining and the collected RNA. Next. We had initially planned to ship the fabricated tissue to Kennedy Space Station Center in Florida by FedEx overnight shipping. However, our shipping test showed that there was an unacceptably high failure rate with regard to the tissue integrity. So we instead opt to hand transport our tissue to Florida by flight travel. Next. Yeah, so this is our chamber and boxed to be carried over to the Kennedy Space Center at the Florida. Next. So once our engineered heart tissue had arrived at the Kennedy Space Center, they are maintained throughout the pre-launch period, after which final tissue quality check and chamber integration and hardware and software check was conducted prior to handover. On March 5th, early this year, our experiment was handed over to be packed securely away in marine unit, which was the power incubator that would keep our tissue warm and while they made a trip up into the space. In this picture, you can see one of our PHAP being strapped in, and entire marine unit would eventually be bolted into the SpaceX Dragon 1 capsule. Finally, shortly before midnight on the March 6th, we was able to see our experiment launched aboard the SpaceX CRS-20 lift supply mission. So this is a real-time movie of the SpaceX 20 launching, which was taken by our science team, which was very exciting moment. Next. So this is a picture of the space astronaut Jessica Meyer during the engineered heart tissue chamber media change and fixing preservation in the life science glove box aboard the International Space Station. And interestingly, Jessica worked with Peter Lee, one of our co-investigator of the science team, in the same lab during her final undergraduate year at the Brown University. What a small world. Next. Yeah, we are excited to find that our engineered heart tissue survived this launch, and they produced the contractile functional data during the 1 month in space. And while the analysis of this data is still preliminary, interestingly, we found that there is already a functional deficit due to the microgravity after just 4 days in space, as you can see in this figure. Next. And the twitching force decreased continuously over time, and difference in beat interval variation, which we use as a metrics for the arrhythmia, irregular heartbeat, show a peak at the day 19 in this figure on the right, but the reduction by day 26 and recovery to baseline upon the return to the 1G, which was interesting. Next. So to summarize, we was able to use our novel electroconductive hybrid hydrogel to improve the functional maturity of our engineered heart tissue, and then by modifying our tissue platform to not only enable automated pulse sensing, but to also be packaged for the space flight. We successfully delivered the experiment to the International Space Station early this year, March, and analysis of the space flight contractor data is still ongoing. We are also currently conducting the ground-based control experiment, as well as the RNA sequencing analysis, which is already delayed due to the COVID-19 associated challenges. We also began to work on the second phase of our project, which is to evaluate whether therapeutic intervention could help ameliorate microgravity-induced cardiomyopathy. So we'll be working on this topic for next 2 years. So we demonstrate that our proposed engineered human heart tissue research model could be potentially serve as an effective tool for better understanding of the cardiac function in response to the external factor such as microgravity, which will be useful for drug development and other applications related to the cardiac dysfunction on the Earth. Next. This is our engineered human heart tissue in space team, which consists of the researchers from the Johns Hopkins University, University of Washington, and Brown University, as well as the BioServe Space Technology, who was our hardware instrumentation partners. Next. Finally, I'd like to thank our funding sponsor, NIH and CASES, as well as the team at the NASA and space that helped send our payload to the International Space Station. Thank you so much for your attention.
Thank you very much, Dr. Kim. And thank you for all the questions that are coming in. As a reminder to those that are just joining, we will be getting to those questions at the end of all the speaker sessions during a panel session. So next up, we have Ms. Stephanie Countryman. Ms. Countryman is the Director of BioServe Space Technologies and a Research Associate within the Ann and H.G. Smead Aerospace Engineering and Science Department at the University of Colorado Boulder. As director, she leads a wide variety of activities, including programmatic oversight of space life science investigations and engineering projects. Recently, she has been involved with experiments examining microgravity effects on proximal tubule epithelial cells of the kidney, induced pluripotent stem cells, mesenchymal stem cells, cardiomyocytes, and endothelial cells. Each of these experiments seeks to utilize the microgravity environment as a model for different types of human disease, including aging, in order to seek more effective treatments for people on Earth. Stephanie, please feel free to start when you're ready.
Thanks, Mark. So today I'm going to talk about going from benchtop to the International Space Station. So essentially, how do we translate a ground-based experiment into a spaceflight experiment? And specifically utilizing one of the microphysiological systems platforms. Duck Ho did such a great job at talking about the engineered heart tissue platform that I'm going to switch gears and talk about one of the other platforms that we also supported. Of course, I could talk all day on this subject, but I'm going to try to cover top-level details in the next 20 minutes. Just quickly tell you a little bit about who BioServe is. We are one of what NASA and ISS National Lab considers one of the implementation partners. We were established at the University of Colorado in Boulder in 1987. We have about 20 to 25 full-time employees who consist of faculty, staff, engineers, and students. We have extensive experience launching life science experiments to space. We flew our first experiment in 1991 on board the Space Shuttle STS-37. Since that time, we've launched experiments on 72 spaceflight missions, and we've had facilities continuously on board the International Space Station since 2001. In just the last 2 years alone, we've supported 15 different complex space-based cell culture experiments, including microphysiological systems and organoids experiments. And by the end of this year, we have 4 more on the books. So I'm going to jump right into talking about translating experiments from the ground to space. This right here is just a picture of a typical cell culture lab or wet lab, labs that you'll see in lab facilities, pharmaceutical biotech companies, science labs, and universities around on Earth. And this is what our science lab looks like on board the ISS. This is the Destiny lab, and this is where the majority of the U.S. experiments are conducted. And you can see that there's quite a bit of equipment in this lab, and a lot of this equipment does support different types of life science experiments. There's even equipment that looks just like it does on the ground. This is on the right side is a picture of Anne McLean using a microscope to do some imaging. In terms of equipment, here are some of the pieces of lab equipment that are necessary that are utilized on Earth for conducting cell culture experiments. We have a biosafety cabinet, which is designed not only to do sterile work, but protect the scientist who's doing that work. We have incubators, 37C, 5% CO2. That's how we conduct our cell cultures. We use, with MPS platforms, we use syringe pumps, and this is what that may look like in the lab. We have -80 freezers, -20 freezers, 4C refrigeration. There's pipetting that occurs in the lab. Rapid freezing is done with liquid nitrogen. We have water baths and centrifuges. So these are all types of equipment that you may see in a standard lab. This is what this equipment looks like on board the ISS. So up here on the left, we have the equivalent to the biosafety cabinet on the ground. It's called the Life Sciences Glove Box. And this is designed to protect the crew and the vehicle from any organisms or liquids that could be dangerous to the crew and vehicle. But it also allows us an opportunity to use a space that is not necessarily completely sterile, but as sterile as we can get on board the space station. We have -80 freezers. It's called Melfi. This is the crew with the -80 freezer. These 4 round doors are the -80 freezer space. We do have, BioServe has an incubator up on station. It's called Sable. It's these 2 units right here. We currently have 3 of them on board. They provide the 37C and the active 5% CO2, but they can also provide a temperature range of -5C to +43. In terms of a lab bench, if you're doing non-sterile work, we have the multi-purpose work area right here. We have a thaw system. That water bath that you saw, obviously we can't have loose liquids on board the station. So we developed a thaw system that is equivalent to a water bath, but it's designed for use in a microgravity environment. There's also a microscope and imaging system that's right here. This is a crew conducting a protein crystallography experiment. There is a centrifuge on board that's primarily used for spinning blood from the astronauts, but it can also be used in cell culture experiments. Just recently, a rapid freeze piece of hardware was designed and launched to the space station, which will be the equivalent of, or similar to, utilizing liquid nitrogen. Up until just recently, this was not available. And then in terms of pipetting, if you're using liquids that aren't hazardous or small volumes where you can use surface tension to maintain those liquids, you can do standard pipetting. But at times when we have larger versions of pipetting, we have to use syringes in an enclosed loop system in order to do that. So how do we translate to the ISS? Well, there's lots of things to consider. So we can consider whether we want to do a manual experiment, which you saw some pictures in the previous slide of the crew conducting experiments manually, versus automated. And there's pros and cons to both types. The number of samples, sometimes with automated systems, because of our limited space, we can have a lower number of samples than the manual. Though, depending upon the processes and procedures that we use for those manual systems, that may also limit the number of samples that are available. Teams need to consider whether they want to launch their cells live or frozen. And that may be dependent upon whether your cells are adherent or suspended. Obviously, if you launch adherent cells, say in a cryovial, if you want to thaw them in orbit and then seed them in orbit in a cell culture plate, that can be tricky because there's no gravity to help those cells settle down to the bottom of the plate. So it's very likely that adherent cells will not adhere if seeded in microgravity. You also need to consider the number of days that it takes for your experiment to get to orbit and then be accessible. You have to think about launch effects, G-forces and vibration, your launch orientation, whether you have requirements for active CO2. You also need to think about thermal control and the uniformity of your thermal control. Automated systems often have electronics in them that generate heat, and so that can actually make hot spots within your system. So managing thermal control is very important. And then in terms of returning live or frozen or fixed, those are also things to consider. And essentially you have the same things. You have to consider CO2 if you're coming back live, and thermal control, and then the landing effects on your live cultures, or even some of your fixed cultures if you're looking at morphology. So you want to consider launch orientation, and then of course the number of days until access to those samples, since it can be a few days from when the capsule unbursts with the station to when it lands in the ocean. You also have to consider bubbles and bubble management. Obviously, the bubbles don't rise in space, and so they can sit right in the middle of your culture, and that can impact either your fluidics or the morphology of your cells growing. And then the temperature control of your samples and medium reagents as they're being transported and returned from the station. You have to consider human factors, how the crew will follow procedures to either power up your powered payload or to complete your manual operations. There's obviously power issues and concerns that you need to address, and then of course material compatibility, gas permeability, and sterility with the unit. And also you need to consider processes that you do in the lab that actually utilize gravity how do you translate them to the spaceflight experiment? So I'm going to talk quickly about a specific example of an MTS device, which is the Nordis Bio triplex chip, organoid chip, that we designed a syringe pump system for and launched to the station on SpaceX 17. So this experiment was called the effect of microgravity on the structure and function of proximal and distal tubule in a microphysiological system. The PIs were Jonathan Himmelfarb, Ed Kelly, and Catherine Young at the University of Washington, and it was sponsored by the NIH, NCATS, ISS National Lab, and NASA. So the requirements for this experiment, on the right here, you can see the Nordis triplex chip. So each chip had 3 samples on it, and each chip had both an influence, an input, and an output. There were 24 chips that the team wanted to launch, so that was a total of 72 samples. The experiment required 37 C, 5% CO2, and high humidity to keep the samples from drying out, since these chips have very low volumes of fluid. They did need a correct launch orientation. The cells were loaded in, the cells formed little tubules in the chips, and they needed those tubules to maintain their shape during launch. So we did have to consider launch orientation. The media that was not used during the transport of the system needed to be held at 4C. The system required continuous perfusion of the media from when they were loaded into the system until the samples were terminated. And that perfusion rate was required at 750 microliters a day. There also was a requirement to collect the effluent from each individual sample and then store that effluent at -80. And then at termination, the samples were either fixed or preserved. The perfusion rate of the fixative and preservation reagent had a different rate of 1,440 microliters a day. So the experiment needed 7 days of microgravity exposure, and it required 2 days of microgravity exposure for the treatment media. It had acclimation media and treatment media, and it also required 4 to 24 hours of fixation. So what are the things that we needed to adapt for the spaceflight system? Well, there were 4 different use configurations. There was the launch configuration, the on-orbit configuration, both inside the stable incubator and inside the life sciences glove box. Because this was a semi-automated system, it required some input by the crew. And then there also was a return scenario. So as we said, we continually perfused the samples until they were terminated. We needed temperature control through all of these phases of the experiment. We also needed adequate 5% CO2. So we did have static CO2 during the launch, and then once it was inserted into the stable incubator, it was active. The treatment media was kept at 4C both on launch and during on-orbit stowage until use. The fixed samples themselves were stored at 4C on orbit and then kept at 4C during return. The preserved samples were kept at -80 C on orbit and then returned at -80. We also had to consider the NASA required levels of containment. So for the cells in the effluent, that required 2 levels of containment to meet NASA safety guidelines, but that containment needed to allow for the appropriate gas exchange so that the cultures could get the required CO2. The formalin cassettes needed 3 levels of containment because that's considered a hazard level 2 from NASA, which requires 3 levels of containment. And again, as I mentioned, we need to think about thermal control throughout all phases and then the human factors. One of the other things that we had to consider was the material biocompatibility. So it's very important for the materials of these systems like this not to impact the science. So in fact, every single piece of material that came in contact with the science, we sent to the PI, and then they did testing in Petri dishes with their cells to ensure that those pieces of material did not impact the science. That material also has to meet NASA safety requirements in that it cannot have significant off-gassing or affect the crew in any way. The material obviously needs to be able to be sterilized, and it also needed to be able to handle -80 C to +37 C. The whole system itself in addition to the science, needed to withstand launch vibrations and G-forces since we were launching live cells. And then once the system was on board ISS, we needed to have real-time telemetry to ensure that the system was working correctly. But we also needed to record all other times that the system was not inside stable so that we could know that the parameters were within normal and that we didn't have any issues outside of the time that it was inside the incubator. So all of this needed to fit within a compact volume. So we had one launch locker to fit all of this, the 72 samples and all the supporting hardware for that. And then we had 2 stables on orbit. So for visual sakes, this is what, if we had 39 samples in the lab, this is what it takes to support that using a syringe pump system. So 72 samples would require 8 syringe pumps on the ground, 4 incubators, 24 large Petri dishes to hold those triplex chips so that they did not become contaminated, 216 small Petri dishes for all of the effluent that would be collected, 216 syringes for media and fixative, and 216 feet of tubing. Just so everyone knows, the Nordis Bio chip system does have a pneumatic system as well that takes up a little bit less space, but it still has quite a high volume. So our challenge was to take all of this, meet all of those requirements, and get it into a system that would now fit into the space flight parameters that we had. So visually, we needed to take 48 cubic feet that we used to support this experiment in the lab. And for launch, we had 2 cubic feet. And then on orbit, we had 1.6 cubic feet. So how did we do that? Well, we did a significant amount of research and development, obviously, in determining how to develop this system. There was a significant amount of testing that was completed both on the engineering side and the science side, and then the 2 sides working together to test the system in the engineering system to ensure that the design was supporting the science appropriately. In the end, the whole system and all of the supporting hardware required to support the protocol, we machined 2,500 individual pieces of hardware. So to show you quickly, the housing, so for the triplex chips, because it contained human cells, we needed to put that in a level of containment, 2 levels of containment. So we developed a housing. On the left picture, you see the square box and a valve block, which is in the right hand of the person in this left picture. And the 3 chips, each housing held 3 chips. And this housing was completely sealed. And on the right, you can see it's fully loaded, this housing. And this housing and valve block system would then interface with the media and fixative cassette. So here we have the media and fixative cassettes integrated with the housing. So the housing provided it with 2 levels of containment. It was able to provide gas exchange for the cell cultures, but it kept all of the liquids contained if there was any leaks. And then the media cassettes, which had 9 channels, because we were supporting, each housing was supporting 9 samples, 9 channels of media, and then 9 channels, 9 individual bags collecting the effluent from each individual sample. This is the syringe pump itself. So 2 housings with their associated media cassette would fit within the syringe pump, and the syringe pump could purge, it could run. The run was when we were doing media at 750 microliters a day. The fixative was the 1440 microliters a day. And then of course, we needed to retract the pistons that you see here in order to pull the samples out of here when the crew needed to do a media exchange. So just to remind you, this is what it looked like in the lab. And in the end, this is what it looked like, but we had 4 of these. So we were able to reduce that down to 4 of these. One of the syringe pumps, and then there's 4 loaded within the launch locker, and then it's sealed up and powered and ready to launch while it's on the Dragon and its capsule, and it's being powered continuously through this entire transport. This is what it looked like on orbit. So here are the 2 stable units. Here is the inside of the stable units. There were 2 syringe pumps and 1 power module inside each stable unit. So for a total of 4 syringe pumps and 2 power modules. And then this is what it looked like inside of the LSG when the crew was getting ready to pull these out to do immediate exchange. The LSG could hold 2 of these syringe pump systems at a time.
Hey, Stephanie?
And then this, yep.
Sorry to interrupt. I'm going to ask that we potentially, okay, now your slides are moving forward. So we see your launch configuration now. So, okay, please proceed.
So I'm
okay,
behind,
now,
where am I? Am I on return configuration?
You're on return configuration now. Thank you.
Okay, did everybody see this one?
Yep, that's coming through now. And now
okay,
LSG and orbit configuration.
Okay, so I don't know if people saw this. So so I'm not sure what everybody saw. So this was the on-orbit stable configuration, and this was the LSG on-orbit configuration. And then this was the return configuration where we only brought the cassettes and the effluent cassettes, and then of course the sample, the culture samples back. Some were brought back at 4C and some were brought back at -80. So the challenges to supporting any type of experiment that's complex like this, that's a microphysiological system platform or organoids even, is reducing to a small footprint, understanding fluid flow and management, understanding fluid flow and managing that fluid flow, and particularly with regards to bubbles. It's challenging to create the appropriate levels of containment, but keep gas permeability at a level that is amenable to your cell cultures. Obviously, we use quite a few plastic components. A lot of plastics are very biocompatible, but machining those plastic parts is very complex, and the tolerances on our plastic parts were very small, and so that was very challenging. Obviously, getting the correct interface between the chips and the cassettes so that you could feed the appropriate media to the chips. Loading quickly and efficiently, designing a system that you can load quickly and efficiently with the science minus bubbles, but keeping appropriate temperature control is very challenging. Keeping sterility during all of the loading and handling, managing the effluent, and keeping everything uniform, and obviously designing a circuit board that is conducive to small spaces. These are all challenges that are present when you're trying to translate experiments from the ground-based to the space-based, but can be managed and met successfully. So my words of wisdom in all of this is that, as I said, it's challenging, yet you can have great success if you plan appropriately. I would say that you need to plan for an adequate timeline. There are times when rapid timelines force innovation, but also due to the complexity of translating from ground-based to space-based, it can also make room for error if you go too fast. I would say that your science and engineering teams need to work really closely together so that the results that you get with your science are due to microgravity rather than a hardware configuration. I would say, I think as Jean talked about, try to plan for multiple flights. It's hard to get things done 100% correctly on the first flight. So if you can have multiple flights, you can address issues and concerns and on subsequent flights. And then of course, you can never do you can never do too much testing. It's very important in this business to test as much as possible, both the hardware and the science, and then the science and hardware together. So that's the end of my talk. Thank you. I'd like to recognize again the NIH NCATS, ISS National Lab, Nordis Bio, the UW, and then of course all of the science teams that we work with. Thank you.
All right, thank you very much, Stephanie. And just as a reminder, please continue to ask your question in the question feature, as we'll collect those for the panel session at the conclusion of our final talk. We'll also work to get to answer all the questions that we don't get to in the panel session and make those available at a later time. And our final speaker for today's session is Dr. Lucy Lowe. Dr. Lowe is a scientific program manager for the NIH Tissue Chips for Drug Development Program at the National Center for Advancing Translational Sciences at NIH, where she manages up to 50 disciplinary team funded by the NIH to develop tissue chips, or organs on chips, potentially transformational tools for drug development. This includes all 9 of the Chips in Space teams. She is also the NIH and HHS NASA liaison point of contact, working to discuss and facilitate collaborations between NIH and other agencies and NASA on areas of overlapping agency interest. Today, she'll be providing an overview of the tissue chip program. Lucy, please feel free to begin. We can see your slides.
Okay, excellent. So good afternoon, everyone, and thank you so much for the invite to talk on the panel today. So I'm going to be shifting gears a little bit because I'm going to be talking about how NCATS and the work that we do, we push acceleration and we push science by, in large part, due to leveraging partnerships. And I'll talk a little bit about the program. I'll also talk a little bit about some of the Chips in Space projects that you've already heard a little bit about today. So just to kind of give an overview of what I'll be covering. So just who we are at NCAT, the tissue chip program, and then one of the things ISS National Lab asked me to talk about was how we work with pharma and the FDA to expand the use of tissue chip technology for drug development. So who is NCATS? We are the National Center for Advancing Translational Sciences. We are one of the 27 institutes and centers that make up the NIH. And we're a little bit unusual compared to some of the other institutes because we are disease agnostic. So you might have heard of some of the other institutes, for example, the National Cancer Institute. It's agnostic. We work very much on the process of translational science to transform the translational process so that new treatments and cures for disease can be delivered to patients faster. So what that means is that we work very closely on a number of things, but include drug development. So leading on, and I'll talk a little bit about the Microphysiological Systems Program, or tissue chips for drug screening. So we've already heard about some tissue chips and some organoids this afternoon or this morning, even depending where your ticked over, but we've already heard from a couple of teams this morning who are doing tissue chip work. So tissue chips, as you know, mimic the biology and the structure and function of human organs and tissues. So the NIH tissue chip program was started back in late 2011, early 2012, and was initially focused on creating platforms and systems that could be used in drug screening. So really working on understanding safety and efficacy of drugs as they go through the drug development pipeline. So yes, the program started in early 2012 and is still going on, but I'll explain a little bit about that in the second. But to begin, this was a 5-year program that was very much focusing on developing these resources and then testing them with reference sets of compounds to really see how they respond. And one of the critical parts of this program, right from the very beginning, has been that we have involved stakeholder end users. So different pharma companies, we've had agreements signed with them so that our developers have been able to get access to proprietary compounds, for example. But also importantly, we've been working with FDA right from the very start. So the idea was that if tissue chips go through development, they're going to be used by the pharmaceutical industry in the end. And so the pharma industry is going to be pushing that data that they're getting to the FDA. So it's very important that the FDA knows exactly what a tissue chip is and what their role is and how they can be helping throughout the drug development process. So we've been very close partners with Tissue Chip. They've been very involved partners all the way along this program. So moving on from 2012, and now here we are in 2020. So the program has really blossomed in the last 8 years or so. We've moved on from the tissue chips for drug screening program. We now have programs for disease modeling. We have the tissue chips in space program. We have tissue chip testing centers, which I'll talk a little bit more about. We have some very specific programs which are focusing on modeling nociception, addiction, and overdose, and also the immune system on a chip. And we have recently just begun a clinical trials on a chip program, which is very much focusing on using tissue chips within the framework of clinical trials to help with decision making. So one of the ways that we have helped expand this program is really engaging and pushing everyone right from the very start to be talking to each other. So this is just a representation. It's actually a little bit outdated, but this is a representation of some of the people that we have involved in the consortium, which is all of the funded teams under those programs that I just showed you. And so while this is, you know, quite a pretty little set of bubbles, what this actually looks like when you start looking at how teams is an absolute mess because everyone is talking to everyone, but that's really what we want. So this slide is, the more that there are discussions going on between our partners, then the better the outcome, because that enables leveraging of resources and expertise, and that's really helping push the. So a slightly easier slide, but it's really just also with external partners outside of NIH and outside of the Tissue Chip Consortium to help pull in people who might be interested to them about how it might be useful for their organizations, and start building relationships and building opportunities with lots of other people. So we have a number of different institutes and centers at NIH that have now started up their own tissue chip programs, and there's some really exciting and very specific organ specific and tissue specific projects that are currently funded by the organizations within HHS, which is Health and Human Services, which is the overarching agency that NIH to talk about how they make chips and discuss state of the science, discuss where there might be interest with other agencies. We've also been working closely with or monitoring what's been going on globally. So in Asia and Europe, that are both starting up many of their own organ-on-chip and tissue chip opportunities, as well as with the US, for example, as I've mentioned, the FDA, working closely with the IQ MPS affiliate, which is actually a group of pharmaceutical industry representatives that actually get together and have created essentially an affiliate where they are actively exploring where tissue chips might fit in the framework of use within pharma, and how we can partner with them. So we're really excited to be able to continue to engage with them. So other ways that we've been pushing and building confidence in tissue chip is very much building a framework that we see is going to help along this path to adoption and commercialization. So that's important for NCATS, because everything we do is designed to help disseminate technology to a broader audience, and that's very much always been the plan with tissue chips. So one of the ways that we've been doing that is actually working on this framework. And so the first sort of step of that framework that we see is the physiological aspects that need to be taken into account when talking about tissue chips. So here we're talking about creation of tissues, the structure and function of tissues as we would want to see. I refer to using training sets of reference compounds, and so we see that this responsibility is really with a few years, and we've been very excited to see the progress they've made. So in the middle of that slide, the number 2 level, we've been talking about analytical validation of tissue chips. So I'll talk a little bit more about the tissue chip testing centers efforts that we have been pushing to help push that part of it. And then we really see that the top level, the number 3 level here, is industrial. So this is really talking about applied use of tissue chips within an industrial aspect. And so, for example, their use within a CRO type environment. And so I'll talk a little bit about that as we go on. Next slide, please. So talking about levels 2 and 3 of the previous slide, NCATS has been funding what we call the tissue chip testing centers, and now actually the next gen testing centers, who are independently funded centers that are, their remit is to onboard tissue chip technology and to validate it within their labs. And so what that means from our point of view is that they can onboard the technology, they can recreate the tissues and the systems within that within their labs in the same way that the developers have designed them to be used. And then they can start testing drugs and compounds and disease modeling as well within these independent centers, as a way to really prove that the tissue chip technology is not only transferable between sites, but also that the systems perform as they are designed to perform. So we have funded 2 tissue chip testing centers, Javelin Biotech, which is a spin out from MIT, and Texas A&M has created their own tissue chip testing consortium. And so this is a list of some of the tissues and systems that they have been onboarding over the previous few years and testing within their organizations to really try and pin down exactly what these systems are best at, what kinds of things they can do, and how the technology is best suited for particular questions. Next slide, please. So unfortunately, now that I've lost the animations, but I've also wanted to point out that we've also been funding what we call the MPS database, which is a centralized data hub that is designed to collect all of the information that is coming from the tissue chip testing centers and is being made available to the public. So the link is down there at the bottom. Anyone can create an account with the database, and they'll be able to see the data that is coming out from the testing centers. It's on tiered access. And a really key part of this is that we've had our partners at the IQ Consortium and the FDA have been enabled pre-competitive access to that. So through a series of legal paperwork that they've been able to sign, they've actually been able to see that data, which is very important for them when they're deciding whether or not to use microphysiological systems within their organizations, and for the FDA to really start seeing how the data is variable, how robust it is, what the best context of use is for each kind of tissue chip system, and how they can get involved with using the data and using the systems themselves. Next slide, please. So I can skim over this now because we've pretty much already covered it, but I was just going to talk a little bit about the Tissue Chips in Space program. So our Chips in Space program is a joint collaboration between the ISS National Lab, the National Institute for Biomedical Imaging and Bioengineering, and NCATS. Next slide, please. And we've already covered how we've had a couple of the teams talking this morning, but our aim really was to understand human disease and biology that would be really difficult to understand here on Earth, and using microgravity as a tool. Next slide, please. And that we had both scientific and technical benefits that we saw from that. And I don't need to talk about this anymore because Stephanie's slides already amply covered that. So next slide, please. So just my last couple of slides here. So one of the other key things that we find has been really important has been connection with commercial entities that are involved in developing and marketing organ-on-chip technologies, or MPS tissue chip technologies. And what's really exciting is that many of these companies are spin-offs or startups from academic institutions that are involved in the Tissue Chip Consortium, which was that bubble slide with all the red lines all over it that I showed earlier on. And so we've really seen an explosion, not just in within the consortium and within academia, but also on the commercial side of things as well. And this is really important because we want these companies to be successfully marketing their products and keeping us up to date and keeping us involved as they go along with that. Next slide, please. So this is my final slide. NCATS has worked really hard on developing many partnerships throughout the years to work together with other NIH institutes, other government agencies, a number of different non-profit organizations and commercial entities as well, and that we see lots of different opportunities for use of tissue chips. The Chips in Space program is going to be instrumental in helping develop the technologies that could be useful for some of those things. And we're really excited to see what the future holds here. And I think I've just clicked to the next slide, and that's my final slide. If anyone has questions, they are welcome to email me. I'm happy to share the slide deck so you'll be able to see the slides better. But yeah, that was it from me. So back to you, Mark.
All right, great, Lucy. I appreciate you. All right, so with that, for the time being, let's open it up. So could I have Stephanie and Jean and Yoko join for now? Okay. So I'm going to start with some of the specific questions that were asked to each of you, and then maybe get to some broader questions. So Jean, and maybe I can expand this to the rest of you after, is when you're planning your flight experiment, how do you handle the gas exchange for your cryo vials? What type of preliminary experiments are you doing? Did you test for a high cell survival at 30 days? Can you speak also to some of the pre-work that you do going into the flight to determine all those conditions?
Yeah, so I should tell you that nobody in my lab believed that those cells could survive in cryovials for 30 days. And we had to prove that. I had to prove it. I'm the one who's not in the lab, but I remember graduate school, and there are a lot of things you can do with cells that the people who've been trained more recently are probably not aware of. So we did not require any gas exchange during the time of the flight. We used an alternative buffer system for the cells. We tested them ahead of time, and actually what you saw in that one of my slides was the electrophysiology. Those were organoids that were grown for 30 days before we did the electrophysiology. So we showed on the ground before we launched that the cells survived really well for 30 days. They were at 37 degrees, but otherwise you don't have to touch the tubes at all.
Okay, great. And Stephanie and Yoko, if you don't mind adding a little bit about how you optimized from your terrestrial experiment. I know, Stephanie, you talked about a lot of the different experimental design parameters that you look at, but Dioko, did you have any specific challenges that you encountered in working towards that? And I think you're on mute.
So prior to our tissue chip in space project, we had the prototype in-house. But one of the challenges for tissue chip in space project, we had to custom, change and modify and optimize for, PHAB chamber, and we have the electromagnetic sensor to measure the contractile force. So we also consider whether there'll be any, radiation in space, interfere with our electromagnetic sensor. So we had to go through all those, potential troubleshooting experiment, and we was actually surprised by the fact that we was getting the signal from the space, and we still analyzed those data, but there was some space instrumentation related challenges. But here, Stephanie, the director of the BioServe, has a lot of experience, so we work very closely each other and have a weekly meeting online for hour and discuss about all the virtual potential pitfalls and how we can overcome those problem. We're still trying to address some of those technical challenges, but I think we have great team to address those challenges.
Okay, great. Actually, while we're on Dioko, can I ask you a little bit more about your system? These are some of the questions that were asked. Does the twitch force measured in the ECM matrix a sign of increased rigidity or stiffness? And I'm going to ask a few more questions while you're at it so you can talk about it. Do you think that some of the functional defects are because of the altered metabolism of the media components? And then can you also talk about how you might determine whether or not the phenotype response that you saw in your experiment matches either astronaut health or for this program, a terrestrial phenotype?
Good question. So first question, whether there is any metabolic changes and that also potentially regulating our phenotypic differences. So simple answer is we do not know yet, but we are hoping that our RNA sequencing analysis could potentially inform what specific pathways involved in this space related microgravity induced cardiomyopathy, and probably metabolic pathway might be the one of the major pathway involved in these phenotypic differences. But we still analyzing our RNA sequence data, so I hope to be able to address this question more data. And another question is this was one single launching experiment, so we need to gather the more data to demonstrate whether our in vitro human heart tissue platform can potentially recapitulate the space astronaut condition. But interestingly, some of our preliminary analysis showing that there is a decrease in the twitching force over time, and there is some arrhythmogenic irregular heartbeat behavior, which is kind of anticipated in the space. So that was an interesting finding, but we are still comparing that with our ground control sample.
All right, great. And Stephanie, can you talk to whether or not when you're doing an experiment or preparing for a launch for a specific science payload, how much of that engineering is translatable to other projects that you might be working on? And while she's answering that, if I could get the rest of you, including Lucy, talk about how maybe the engineering or other scientific observations made were translated to terrestrial type of experiments.
Sure. So So in terms of translating the engineering to other experiments on the ground or in space?
For you in space, and then if you have the applications for terrestrial use, that would be great too.
Yeah, sure, definitely. I mean, supporting an experiment in space and the engineering that we do has really been a culmination of experiment after experiment after experiment. You know, I've been honestly in the business for 20 years, and I still learn something every single mission. And so a lot of our engineering that we develop for one payload is definitely applied to the next follow-on experiment. We may have to do modifications to the hardware to support the particular experiment, but a lot of the principles of the engineering that we support are translatable from one experiment to the next, especially like Duck Host experiment is one example where the PHAV, that aluminum box that we use to transport the science to the space station, but we also use it as a level of containment, and it also provides gas permeability. So that's used, we've used that probably 30 or 35 times on different spaceflight experiments, different cell culture experiments. And then from the ground, one of the things that it does, and particularly for the NPS platforms, is that figuring out how to get a system that takes up so much volume in the lab into a small footprint is translatable to the companies, and particularly Nordis Bio in this instance, where they're trying to make their system more user-friendly in the lab in terms of space. And so they're actually utilizing some of the principles and engineering concepts that we use to develop that system to now develop a system for lab, in the lab on the ground, that has a similar footprint. It obviously won't have the levels of containment that are required for a spaceflight experiment, but they're trying to match that same footprint. So some of those principles are applied to ground-based research for sure.
Sorry, I'll open it up to the rest of you, if Lucy, or if you've seen other programs that have developed systems that have helped translate to, from the engineering side, have helped advance the field, in the MPS field or an organoids field.
Yeah, exactly. Can you hear me okay?
Yep.
Okay. So, yeah, I feel partly responsible for some of Stephanie's stress related to some of the spaceflight experiments she's had to do over the past few years, because Stephanie and Ducko are both part of our Chips in Space program, which I was going to mention on the slides, but now I can talk about it here anyway. So we're funding, NCAS is funding 9 projects through that program to actually send 9 different groups and their physiological systems, microphysiological systems, up to the space station. And both Ducko and Stephanie did a fantastic job of explaining what that is and also why that's really difficult. So Stephanie specifically mentioned tight deadlines, and that's definitely our fault, as well as being on a launch schedule. So her and the team at Bioserve have done a really incredible job of, as she explained, shrinking down that technology to something that is robust and reliable enough, and as Ducko was talking about, robust enough to survive launch. And for example, the tissues and the engineering needs to survive, but then has to fit down into this tiny footprint. And so for us particularly, that's really important, because the whole point of the Chips in Space program was about the biology and about using microgravity as a tool to model disease and to understand new ways that tissues might be reacting to microgravity, so that we can use those opportunities to look at different drug opportunities, different therapeutics, but also for the technological aspect of it. And that's really critical from much less so than the 48 square feet that are needed, for example, in the lab to begin with. But that makes it much more translatable down here on Earth. It makes these systems much more usable for a broad, unskilled user base back down here on Earth. And so really the Chips in Space program, from our perspective at NCATS, has been instrumental in pushing the technical advancement of MPS and organoid systems that will be directly translatable to those broader use of those systems down here on Earth. And I think that's something that pharma and FDA have been very interested in and have been following closely.
Dioko or Jean, do you have anything else to add to that point?
Go ahead. I'll talk last. Go ahead.
Oh, you froze him, so you can go ahead, Jean.
Okay, I froze them. That was cool. I didn't know I could do that. Yeah, so you can probably see that we've sort of de-technologicalized our experiments, and that was the result of experience that we had with some of our early tests that that led us to eventually realize that less complexity was better. And I was just struck by the point that, you know, people who are not well trained should be able to do this. And I think our organoid in a cryotube is about as low tech as you can get, and yet really remarkably effective in preserving those cells over a long period of time. We can add some bells and whistles to that going forward, but we've established that in our case, simple is better, and that a brain organoid at least is perfectly healthy and does very, very well just floating in a fluid in space.
All right, great. So I guess one of the questions, maybe Stephanie, you can talk about this a lot, is also for Dioko. So it appears that sending tissue chips or organoids to space might be easier than shipping them across the country. So are there any lessons learned, or is this an issue with the field in general here in terrestrial fields, sending these tissue chip or organoid systems from one lab to another? And have there been any best practices gained from this? Okay, it looks like Jean.
Looks like Jean. I can let
Yeah,
Jean
I was going to
ahead.
say. Yeah, I was just going to say something very, very quick. We discovered that the G-forces on a FedEx truck are greater than the G-forces of the launch.
Or the delivery person chucking it in the back of the truck, right?
Yeah, it didn't. Yeah, exactly. I think at one point the thing fell off, and yeah. So it was a lot rougher being on the ground than it was being in space.
Yeah, so if
But they survived.
Yep. So we wanted to do, you know, we looked into FedEx, because FedEx will say that they can do some shipping tests for you. If you send them their box, they'll do some tests for you to see, you know, how and it would be, the test reflects what an average package will see in an average shipment. So we didn't really have time to do that because it said it would take about a month. So we asked if we could get the parameters for the test they do. And so they sent us the parameters, and the parameters of what a package, what they say an average package would receive, is essentially it would be dropped from 3 feet 10 times on all different axes. Mm-hmm. That's what your package sees during a process. So if you think about that, that's being a package that weighs, you know, 20 pounds from 3 feet. That's a lot of force. And it's not force that they see when they're launched on the rocket. So definitely hand carrying if you need to ship your tissues, you know, fragile tissues from one location across the country is an option.
And Lucy, do you see this as a general concern with the fields when you see at a larger picture is transporting samples from one site to another?
Yeah, absolutely. And I think it's almost funny that it's easier to ship samples to space than it is to ship them cross country. Ducko can attest to the fact that hand carrying is safer. There are a couple of couriers that say they can do it, but that remains to be seen. But this is actually really critical for our tissue chip testing centers as well, and it's something that we've seen multiple times. It really can be the most simple things that can cause the biggest problems. And if that's just getting cells or supplies from one lab to another, it can be really problematic. And so one thing that we've definitely learned, unfortunately the hard way, is that the tech transfer actually is the biggest aspect of any of this kind of development, whether it's organoids or tissue chips or any sort of cell culture system. Tech transfer between labs is so critical to try before you start doing it for real. Sending test packages, making sure that the people are on either end to receive it, just getting right down into the nitty-gritty of the logistics is absolutely critical for sure. And Ducko, I don't know if you want to comment on that. I know you've had some excitement and issues your end over the months, over the years.
Yeah, sorry for network issue. So Stephanie and Lucy share a lot of our experience, and one of challenge in our project was our science team members should have to go through the TSA pre-check to deliver the sample to the from Seattle all the way to the Kennedy Space Center in the Florida. And when we try to deliver our tissue sample second times, actually people in the airport was aware of our experiment because they have seen this. So this is one of the intrinsic challenges in tissue chip in space project.
Yeah, I'm interested in what TSA did because we decided not to hand carry them, even though that would have been a lot simpler for us, because we thought that's all we need is to have TSA ask us what this is and then tell us that we had to throw it in the trash.
Okay, so Stephanie, you are
I should say that we prearranged everything with TSA prior to our arrival, so to ensure that we, well, as insured as you can be, you didn't have issues.
All right, so here's a general question for the use of MPS systems or organoids in general. So, obviously they're not as complex as an in vivo system, and they're obviously more complex than a simple cell 2D system. So, what are some of the trade-offs that you see with using organoids or MPS devices to model disease progression? So, are there things that you have to be concerned to include so that you're recapitulating an in vivo-like environment, or are there things that that you're missing or maybe confounders because of the necessity of the setup? So, I don't know, Dioko or Jean, if you want to start.
Yeah, I'll start. So you notice that we got rid of all the technology essentially, and so we're starting from a ground state in which we can now add on things gradually. We are interested in the engineering of a system in which we can add things in flight. Although I think if you're going to be realistic about what, that would be what you would require if you were going to test drugs on the space station. If we're really interested in the physiological effects of being in space, then we could perturb the cells initially as soon as they get up there, like an astronaut would land on the space station, and then 30 days later we would ask, well, what was the effect of that perturbation on those cells? And I think having specific iPS cells from people with neurological diseases or neurodegenerative diseases was, I think, critical. If we could have used any old cells, but I think just for feeling like we could cover all the bases, that was a really great approach. And so that means that we have the entire iPS cell space around us. We can study any neurological disease in space by creating these organoids, which do become more complex. Obviously, the next thing I want to do is to create a more complex organoid system in which the neurons have their targets there as well, rather than just being there on their own. So I think I hate to say this, but I think the sky's the limit.
So my laboratory research focused on the engineered human heart tissue platform. And there's definitely no trade-off issue. So if we make a more complex, sophisticated model, then the throughput become lower, and the robustness issue and reliability issue also there. But if you make a simple model, then we can make a more reliable and robust and higher throughput. So we need to make some decision to considering this trade-off issue. So in our tissue chip in space project, we decided to use a 24 well tissue platform, which is reasonably higher throughput. And based on our in-house test, our 24 tissue platform give reliable, relatively robust data. So and we are still developing the 96 tissue platform for potential application in the space biology experiment. But we're still trying to optimize the robustness issue and reliability issue.
Yeah, Lucy, do you have anything else to add to that as far as the systems and their recapitulation of true in vivo environments?
Yeah, and I think something that comes up again and again when we talk to both FDA and pharma is this concept of context of use. So every single platform that is created, whether it's an organoid or a tissue chip, is going to have a very unique set of things where it can be helpful. And as they say, all models are wrong, but some models are useful. So that's very much the case with tissue chips. They have very specific areas where they can be very, very useful, and then other areas where they can be less useful. So if you're talking about high throughput toxicity screening, then a more simplistic model might be very, very helpful. But if you're talking about really representing a complex multi-organ disease on a series of linked chips, and looking at the efficacy of a promising therapeutic, then that's where tissue chips could be incredibly helpful. So we see that there's real utility across the broad spectrum in terms of simple versus complex models. We also see a lot of what is being referred to as synergistic engineering, which is essentially organoid models, so spheres of blobs of cells, but on microfluidic platforms, because this actually helps add a level of physiological relevance to some more of the kind of the simplistic organoid models, because it exposes them to fluid shear and stresses that they would be subject to in vivo. So it helps add a little bit of complexity to organoid systems. So we see that there's a lot of utility in that area, and we actually are seeing a lot of that in the field, and some really exciting science come out right now. And Jean, I think you've got some comments there.
Oh yeah, so we started out with a microfluidic platform with medium exchange and the organoids in the little wells, and it was a very sophisticated engineering. And what we found was that we had the most mundane of problems, like the organoid would end up blocking the media flow. You know, it was just, and then we had some contamination issues because it was too complex. And so we really kept going backwards until we came to the simplest possible system. That means that we at least understand already the issues that we'll have if we try to make this more complex, and I think we can adapt that with the engineering so that we don't make those mistakes again.
Yeah, great. Okay, so let me just build on that then a little bit. And what are some of the capabilities, or maybe even an experiment, you can be totally biased, that you'd like to see performed on the ISS, or a capability that you'd like to have on the ISS that would really advance your science or the field in general? Have you any thoughts on that? You want to go for that?
I do. I think stem cells are super hot right now. I mean, everyone knows they are. Stem cell technology is incredibly exciting. Down here on Earth, it's also incredibly variable, and it's very, very difficult to get reliable results, to get to understand differentiation protocols, to make sure that the cells are mature to the level that they need to be useful. And I think it would be really, really exciting to do some microgravity experiments to really try and pin down some of those fundamental aspects that are related to stem cell differentiation and that could really transform the field back down here on Earth. I'm not going to talk about why stem cells are so important. I think you guys are doing a session on it later on this week, but if anyone else wants to weigh in there, then go ahead.
Yeah, I'll add. So we've spent the last 12 years in my lab trying to get to working on reproducibility of differentiation from different iPS cells from different people. And it turns out that just like any engineering problem, that we've managed to solve it so that we can get exactly the same cells from 100 different people using the methods we've developed, which are, you know, we have to do this in a company really, because it takes so much trial and error and so many testing of single variables. But we've accomplished that, and we have an outcome measure that we use, a gene expression profile, which I think you mentioned earlier, Mark. We started with pluritest. Pluritest is a gene expression profile typical of pluripotent cells, and it's predictable that the cells will differentiate into 3 germ layers. We also have another test called neurotest, which is a precise calibration of exactly how differentiated a particular cell type is, at least with respect to neurons. So those problems can be solved on the ground, and I think, I mean, I agree with you. It'd be nice to see whether they turn up again in space, because we feel like we've got our arms around it, but maybe as soon as we start trying something a little bit more exotic, then we'll lose hold of it. But But we had the best possible starting material, and that was really critical for our success.
I also agree with Jen. So quality control and reproducibility of the different cell is very important. And differentiated cardiomyocyte is still not quite mature yet. Even the commercial cell source is not mature. So a lot of the research effort has been made to make a more mature cardiomyocyte for variety of the application. And for space biology experiments, since we have very limited opportunity, this robustness issue and reproducibility issue, quality control issue is more important because there's a lot of biological system noise around this model. So there is a lot of omics technique has been applied for cardiomyocyte maturization characterization. But I personally think the functional maturization is very important. So if we have the more advanced assay, which allows us to have the electrophysiology and contractile force, longitudinal assessment without fixing the tissue, we can still measure the living heart tissue function over time without damaging, without fixing the tissue. So this kind of longitudinal assessment capability will be very helpful for functional characterization and reproducibility and cell variability issue.
Okay, great. So building on that, with issues of reproducibility and maturation and model validation. So, Jean, Stephanie, and Dioko, you guys straddle the commercial academic worlds, and how do you balance the data sharing, the knowledge gain in the fields with trying to make sure that you are able to secure the necessary IP that you need to have a successful commercial business?
Yeah, so I can answer that really simply. You file for patent application, and then you publish the work.
So
yeah,
in our
go ahead, Dr.
Go ahead.
I was going to say that's what happens with our commercial partners. Typically we don't publish until the IP is protected.
So in our tissue chip in space project, we filed the IP first prior to starting this project, and this IP has been licensed to the startup company Curie Bio during the execution of our project over the past 2 years. And a lot of our troubleshooting effort, optimization and reliability checking, those was helpful for also technology commercialization.
Yeah. And if sorry, I was just going to add the purpose of my company is actually to create cells for treatment of people. We're planning to inject those cells into people's brains. So the quality control has been the highest priority for quite a long time. And all the company's efforts are really focused on that. So that's a real advantage for us for our other like little spin out experiments like sending cells into space, that all those things need to be established for a clinical trial. And so that means that all the criteria for release criteria and all those things are being established by the company, and then we get to use the company's technology to do things that are a little bit more fanciful. And sorry, this is just way too much fun, so that's why I call it fanciful. But it is very, very serious.
Yeah, and I guess maybe to elaborate on that a little bit more, is there room for shared information that maybe moves the fields that isn't necessarily that you need to secure the IP for? So, I know that Lucy, with the Tissue Chip Consortium, you really work hard to have data sharing and have transparency as much as you can within the group. So I'm just wondering, in the field, what are some of the best practices you've seen other than, publishing and patent protection, which obviously has a significant lag in time between knowledge and dissemination to the field?
So just briefly about the consortium. The consortium has been going for 8 years now, and it's grown really quite large. but it takes a long time to build the relationships and the trust between people to allow them to share that information. So we have a lot of legal documentation set up that allows data sharing. We have private repositories and databases that people can put their data in. But really, there's a very tangible human aspect in all of these kinds of things, because you have to be able to build partnerships, and often part of that actually comes down to meeting at conferences and actually interacting in person. So hopefully next year we'll all be at ISS R&D in person and be able to chat about these kinds of things over dinner and drinks. But certainly from our perspective, a lot of the ways that partnerships and that data sharing comes about is by interaction over the course of time, essentially, and people getting to know each other, people building those collaborations, talking about the science, realizing that there's shared opportunities and innovations there, and then building their own collaborations. And it's really just a process of time, which sounds kind of fluffy, but that's what we've really seen in our experience.
And NIH Tissue Chip Consortium has been doing fantastic job. So each year there is 2 meetings, spring and autumn. So each PI, share their listen to progress and new data unpublished through the poster presentation or podium presentation. There is also, Slack channel to share some, information and some technical tips. So that has been very helpful.
Okay.
I would add from kind of the engineering and the implementation partner side, we complete a lot of non-disclosure agreements with our commercial partners because depending upon the complexity of the experiment, there are times where the commercial partner has to share a lot of data and information with us that they have not shared with anybody outside of their company. And so sometimes that can, be a little bit scary, I guess, for the commercial entity. But then what we also find is if there's components of what their experiment is or their science is that would be beneficial to another entity trying to fly a space flight experiment, we've had instances where we've spoken with that commercial entity and there are aspects of it that they are willing to discuss with other groups. Obviously, they don't discuss their core protected technology, but we found that organizations are willing to share certain aspects and knowledge that they know in order to help other groups. Obviously, they're not going to share their core technologies, but I think particularly around space flight, there seems to be a lot of willingness to share information.
All right, great. And with that, unfortunately, we're out of time. So I would just like to take this moment to thank each of you for your time and for your presentations. And I hope that the audience enjoyed it as much as I did. We will work to capture all of the presentations and make it available publicly in the not so distant future. And as a reminder, please join us on Thursday, August 6th, for our final session in the series focused on stem cells. So once again, thank you all, and have a good day. Bye.
Thank you, Mark.
Thank you.
Thanks very much, everyone. Thank you. Bye.